So in today's session, we'll mostly look at types of motifs, and basically types of motifs we've already looked at. So we'll study the behavior of these motifs in terms of steady state and alkynes and everything. And you can will I already have given you the GitHub repository where all the JSON codes are there for the all the models that has been used in the code. Please use those JSON codes and the JSON software in general to kind of get yourself accustomed. And so the thing is, in the exam you'll not be provided with any kind of software or anything, and you'll not be asked questions in terms of like very difficult ones which you have to calculate throughout a lot, and then you have to answer. But the idea behind using those plotters and different kinds of softwares for seeing the alkynes and the stability is basically you understand the understand the the characteristics of these motifs and how they behave in terms of stability and alkynes. Okay, so let's get started with the first one today. Okay, so this is a generic example of a mutual repression. So as you can see, S is an enzyme here which is kind of help in generating R, which in turns is positively regulating this dephosphorylation process EP to E. And once this E is formed, E is positively regulating the degradation of R. So basically what is happening is uh, it's a case of mutual repression, because the more the R, the more EP will be converted to E, and the more R will be repressed. So this is an example of a mutual repression, and now we'll see what are the different characteristics for it. So let me draw it here. So this is R here. Now R is positively regulating this behavior, movement of EP to E. So basically this is a dephosphorylation reaction. This is a dephosphorylation reaction. So what happens in biological systems also, you'll notice this activation deactivation is mostly done through phosphorylation. So either you kind of add a phosphate group to a particular molecule, that molecule becomes activated, or you remove it from the particular molecule, it becomes deactivated, and even vice versa. So basically phosphorylation is a major tool that is used in the cellular system and the signaling networks particularly, where basically phosphorylation and dephosphorylation of a particular compound leads to activation or deactivation of that compound. So this is something that you'll see very commonly, dephosphorylation or phosphorylation reaction for activation or deactivation. Okay, so now EP is getting degraded to E because of R. Now E is positively regulating the degradation of R. So this is what brings about the mutual repression network. Now let's try to write the ODs related to this. So dr dt will be one OD, and the other OD is de dt. We want to check these two. These two are main components of this network, and we want to check the characteristic behavior of these two. Just a second. Okay. So here there is an S. Okay. So dr dt will depend on S. It will depend on E and R, basically all of them, right? Because dr dt is getting produced with the help of S. R is helping production of E, and E again is degrading R. So and R is in itself getting degraded as well. So these three are the major major parts of the network that we are looking at. Now DEDT will be a function of R, because DEDT depends, the production of E depends on the production of R, so this will be an R. Next will be E itself, so DED change of E is dependent on E itself. So these are the 3 and the 2 elements of the networks that we are interested in. Now let me try write this down single-handedly. So for R, okay, yeah, so R is positively regulated by S, and if this reaction is going on a rate of K1, so R is positively getting induced by S, so this one will be K1 into S, right, because S is positively regulating R, so positive sign K1 into the concentration of S. Now for R itself, let's term this as K2, and let's let me check once. Okay, let's term this as K2. So R is getting degraded by K2 rate, which is helped by E. So let's tackle R first. So it will be a negative of K2 into R, right? So because R is getting degraded with the rate of K2. So another way to look at this is basically R is activating E, which is degrading K2 as well. So we have to bring the E factor to the equation as well. So we have taken S and R. Now how do we deal with E? So E is how it is contributing to R. It is kind of negatively regulating R, right? Because E is helping R degrade with the rate constant K2, and R on the other hand is helping E to get to increase in concentration. So this will be a negative of K2. This is K2. Negative of K2. No, sorry. Let me try another one. So let's try this one as K2 dash. Right. So what is this part of the reaction? If you notice, what is this part of the reaction? This part of the reaction is E plus R K2 dash. Say X is getting degraded to Y, so it's production of Y. So basically degradation of X. So this is the reaction E plus R, and if you take an X here, E plus R plus X, which is getting degraded to Y. Now looking at this standalone reaction alone, so it will be a K2 dash here into E into R. So that's why I did not write the X part, because X does not have any anything to do with this reaction particularly. It's just E and R degrading. This is already R here, so E and R are degrading itself. R is getting degraded itself to form a new compound. So this whole reaction is given by this equation. So this is the drdt form, right? Now let's try to simplify it a little bit. Let's say K1 into S plus, let's take the minus common here and the R common, so becomes K2 plus K2 dash into E to R. So drdt becomes this, right? Just a simplification of this step to this one. So drdt becomes this. Now let's look at dedt, next part. So dedt. So from here dedt depends on R and E, right? E is getting formed from R with the help of R, and E is also regulating the R compound. So dE dt depends on R and E, but these are like, sorry for this. Okay, so this is a reversible reaction here. If you look at the network here, so let me go back to the network. So this here is a reversible reaction. So this part of the reaction, which you can, sorry, which you can, let me draw here. Okay. So this part of the reaction, if you notice, this is a enzymatic reaction. So this, what will do is it will follow Michaelis-Menten. So this is the reaction here, EP. This is a reversible reaction here, E, and R is the enzyme here, right? So EP is getting degraded to E, and R is acting as an enzyme here. So this part of the reaction will follow Michaelis-Menten constant. So let's okay, so let's go back here. So now, okay, so dE dt here it is this reaction, right? So this reaction will model. So let's assume that this is like a K. No, I'll write it there only. Okay, so let's assume the forward reaction is K1E. Sorry, K1, not E. The forward rate of the reaction is K1, and the reverse rate of the reaction is given by K2, right? So we have already taken K1 and K2. You're saying something?
We already use K1 and
So what
K2.
I'll do is I'll use K1E and K2E. That's what I was saying. So we have already used K1, K2. So we'll use this form of the rate constants for this particular part of the reaction. Right? Now, if you do, let's take the reversible reaction first. So the backward reaction, E to EP. Right? Let's take the backward reaction first. So this will be minus of K2E, because this is the reaction that is going opposite. So dE dt is negative here because E is getting consumed. Okay, so K2 into E. Let me write it properly. Sorry. This is the reversible backward reaction from E to EP. So this depends on R, because R is the one that is regulating this reaction. So R is the enzyme here. So this depends on R, but R, if you take the context of the whole equation, whole model, basically R is not an enzyme. You cannot treat R as an enzyme because the concentration of R is not constant. So R you can assume here as another reactant. So this reaction becomes basically EP plus R, which gives reversibly E, right? And the forward reaction is given by K1E, and the reverse reaction is given by K2. So you can imagine this part of the reaction like this, right? So EP, and you have R, which is getting degraded to E, and there's the reversible reaction with it. So if you apply Michaelis-Menten constant here, so this is your K2 into this is the reversible part. So the Michaelis-Menten constant will be like, it will depend on E, and there will be another Km. Remember, from Michaelis-Menten equation, there is a Km, which is basically equal to the concentration of the substrate at half the velocity. So this was the graph for Michaelis-Menten. So this is given as, sorry, I was telling wrong. So km is the maximum. Oh no, sorry, just, yeah. So this is your maximum velocity v. So the concentration of the substrate S, so this is substrate S, the concentration of substrate S at v by 2, half the velocity, so this is assuming v by 2. So at this point is basically your km value. So this is the Michaelis-Menten equation. So the equation is basically E by E plus, let's put that, K2ME, because this is K2E, we just put the Km value as K2ME, right? So this is this part of the reaction. This is this part of the reaction in a backward manner. Now if you want to do the same, model the same in the forward manner, you add K1E, so this will be E at time t, basically whatever equilibrium you are using, E at time t, but E at time t plus K, let's assume M1 1 ME. So this is the corresponding Km value for the reverse reaction. So the equations, if you notice, it's a little bit different, because here you are on the forward reaction, there are two substrates, EP and R, so you are modeling for both of them, and for the reverse reaction, E is getting degraded to EP. So these two, in the reverse reaction we are not concerned about R, right? In the reverse reaction we are concerned only about the E. So this E and this E are different. This is basically E at equilibrium, and this E is basically the this E is basically the concentration of E at equilibrium, and this E is basically concentration of E at time t, because this E is also getting used up in another reaction. So this E is not in equilibrium with EP. So it is in equilibrium for this particular reaction, but E again is getting used up. So our equations become these two. So this is for dE dt, and this is for dR dt. Now if you want to make certain substitutions and everything, say for example E, you can substitute as E equal to E equilibrium minus E at time t. Okay, so this is the total E given, this is will be a positive, yeah, this is the total E in the system. This is the equilibrium concentration of E, and this is the concentration of E at time t, right? Now if you kind of rearrange this, you can just, for ET you can substitute E minus EQ. So this you can do here. So I'm not doing it, it's just a substitution. You can substitute ET and ET here from this equation. Now what happens to actually the characteristic of this particular network? Let's look at that. Okay, so now if you model this, now if you find the solutions to these particular models, what you will see is, and if you kind of plot the steady state of R versus steady state of S, just to remember, this is R here and this is S. Okay, if you are wanting to model the steady state of R and steady state of S, it depends on the initial value of R, right? If you model this, what you will see is, if you take higher initial values of R, what you get is, let me use another pen. Yeah, what you get is this trajectory. So first is increases a little bit, then there is a jump, and there is another increase, right? So this is a sigmoidal function, as you know. So for high initial concentration of R, this steady state of R is actually sensitive. So say for example steady state of S is here. If you just move from 4, concentration of S to from 4 to 5 here, the steady state jumps a lot. It's here now. So there's a sigmoidal function in play here. So when till a certain concentration of S, it the steady state of R goes linearly, but when a certain spike is in S is given after this particular junction, the steady state of R jumps up. So this is one of the characteristics of a basically of this kind of a system. S and R, if you try to plot, you will find these kind of. So you can use the GitHub page. I'll just once again. This is the GitHub page. I guess you know. If not, use this. So this has all the JSON codes for these kind of models. So for example, if you go here, codes. So this is our basically so for example, reprocellerator, if it's a double negative feedback loop. So all the codes are here. So if you can check it out, so these are your ODEs, these are your initial conditions here, these are your parameters, and what you can do is you can use JSIM, you can use these codes for this particular models, and what you can do is check how the behavior of the basically the concentration of different substrates, how the behavior of the whole network basically is changing with a slight change in like these parameters. And if you remember from bifurcation, if changing of parameters will give you multiple steady states and different types of steady states, it's called bifurcation, right? So you can also check if the system has bifurcation or not with slightly changing these parameter values. So you can see if you're getting another steady state or not, something like that, right? So this is I'll suggest you use these kind of codes and try to model these systems, and this will be a good exercise for you to understand how the systems behave, and you'll get a better idea of them. Okay, so now coming back, yeah. So as I was saying, okay, the concentration of S is in a linear relationship with the steady state of R only when there is a low initial R, right? If there is a high initial R, you have a sigmoidal function like this. Okay, so this was a mutual repressor, right? So this is increasing the degradation of EP to E. E again is increasing the degradation of R. So these are mutual repressors, and this is how the steady state of R and S would look like for different values of initial R. Now let us see. Actually, this was the one that I was showing. Now S is also, so we have three concentrations that we are interested in. So S, E and R, these three are the concentrations, mostly E and R, but since the value of S actually determines the degradation of R and the value of S, so basically here the S is here, so S is contributing to increase of R, and S is also contributing in some way to the decrease of R, because increase of R will lead to this formation of E, which will in turn decrease R. So S is not the thing is DSDT will not exist because this is an enzyme in this reaction. So S, the concentration of S will not change. But if you change the concentration of S, does the equilibrium shift, or does the concentration of R and E and other molecules in the network, how do they change depending on the concentration of S? Okay. So this is the basically the characteristic of how it shows. So this plot I already showed you. So if you have high initial value of R, you have a sigmoidal function. If you have a low initial value of R, you have a linear function. Now if you notice the concentration change of R and time, it depends heavily on S, right? So what is happening is, so basically this is the one sorry, 1 second. Just a second, I just want to check what are the values used for R. So I'll just take a second here. It's not given in my document. Yeah, got it. Yeah, so this is basically I was just checking what are the values of R used, so it's okay. So this is basically the time course of R at different values of S. Okay. So one of these curves is basically showing the the solid line is showing the formation of R. The dotted lines are showing the degradation of R depending on the values of S. Now if you notice one thing, so for example, let's first notice the formation of R versus time. These three solid lines, green, red and blue or black, basically are showing you the formation of R and how it is varying with time. Now if you notice, for S equal to 2 and S equal to 6, these two lines, right, they are pretty close to each other. Like, so what it means is these two will be similar, will have similar steady states. Now if you check for S equal to 12 in the green line, from moving from S equal to 6 to 12, there is a big jump in the concentration of R. It increases much more rapidly than S equal to 2 and S equal to 6. So what you see here, can you tell me what you see here? What is the characteristic that is observed here? This is basically bistability. So at certain points, certain values of S, you have one steady state, which is this one, including these two. And if you increase the value of S to a certain point, the steady states move to a different location. So for S equal to 12, this is another steady state. So if you, I think, increase more from 12 to 20 to 30 and those stuff, basically those will club together. So what happens is you have two steady states here. So at the higher values of S and another at the lower values of S. So this is what basically tells you that this system is bistable. Okay. Yeah. So this is what is telling you that your system is bistable. You have multiple, at least from this plot you can see there are two steady states differently. So the concentration of S is very important because although ds dt will be 0, but the initial concentration of S will affect the values of your steady state. For different values of S, you will end up with different steady states, right? So first, just to recap from what we have seen till now, so first there is this network. It's a mutual repression network. Sorry. Then we checked for different, what are the equations for this network. So we wrote down dr dt and dE dt. And now if you model this, you check the concentration of, check the steady state of R, how it is changing with concentration of S, right? So in also this also depends on the initial value of R. So if you have an initial value high, you have a certain like certain behavior. If you have a low initial value of R, you have another type of behavior, right? And if you plot R versus time, it depends on the concentration of S at different time points. So basically R depends on the concentration of S, and as you can see, there is a bistability in R, if you can see here, because these, if on the lower values of S, they have similar steady states. If you increase the value to after a certain degree, it will have another different steady state. So at least this is a bistable. This might be tri or like tetrastable as well, like you don't know how many stability points are there, steady states are there, stable steady states. But yeah, from this at least you can say like at least two steady states are there, right? Now you try to plot the null client. So going back here, if you sorry, okay, so we put both drdt equal to 0, and we also put dedt equal to 0 for the null clients. So if drdt equal to 0, this becomes k1s minus k2 into r minus k2 dash. See, k2 dash is this one, formation of help how E is degrading R. So this is k2 dash. Just k2 dash into E into R is equal to 0. Now if you rearrange this, you will get a value of S from get a value of R from here, because this is the null client for R. We are trying to find the null client for R. So you take R on one side, so this will be like k1 into S equal to, take R common from here, this will be, sorry, this will be basically taken there. So this is a positive. So if you take R common, k2 from here, and this is another k2 dash into E. Equal to, it's both of them are equal, so equal to here. Now check for R, what is the value of R? You just divide it. So R will be k2 plus, sorry, it's the opposite, right? S divided by k2 plus k2 dash into E. This is the null client for R, right? And if you find the null client for S, it's the same thing. You put what are the null clients we are comparing here? Okay, E and R we are comparing. So for different values of S, you will have different steady states as I showed you. So this is, you can get R, same way you can get E. Remember, you have to substitute this thing in this, but these are like, what to say, mathematical notions. You have to simplify and those stuff. We are more interested in how the behavior looks. Okay, so how will the behavior look? Now if you see, if you plot the E null client with the R null client, these two, sorry, I have to use this one. Okay, if you compare the E null client with the R null client, so this here S is constant, so because for different values of S the R will be different, so here we have taken S to be constant, to be 6. Then you see the E null client and the R null client. Now how many steady states do you see here? I see one here, I see one here, and I see another one here. For a single value of S, you can see there are three what to say steady states, because E and R, these are the ones that we are looking at, and these are their null clines, and where the null clines will meet will be the steady state of the system, right? So this is giving us three steady states. Now the value of R, now we saw that how it depends on S. It also depends on the initial value of R naught and E naught. So basically the initial R concentration and the initial E concentration. The value of the behavior of R depends on both of them. So if you have a higher R naught, lower E naught, you have a behavior like this, the red line, right? This is the behavior. If you have a slightly lower R naught but a slightly greater E naught, actually R is going the opposite direction. So in the first case, with these concentrations of R and E naught, R actually increases with time. If you notice, R increases, and this you can call like a saturation point here. Here is the saturation point, but generally R increases if this set of values are used, and R decreases over time if this set of values are used. Now, like this I am showing you as an example, but how can you find out? So the basic idea would be, if you're doing in pen and paper, you take you have to draw the null clines and check how many steady states are there, and then for different values you have to check. But if you're using JSIM or any other kind of plotter, what you can, there's the loop option in JSIM, if you remember. So in the JSIM model, you can iteratively have different plots for different values of R naught and E naught, right? So you can do this using JSIM. So why I am showing you all this, because the initial concentration of E, R and S here all have a vital role to play in the overall dynamics of the system, in the overall change in concentration of a particular compound in the system. So all these parameters, these are all parameters because these are initial conditions, right? So when you're defining the OD, you'll give different initial conditions. So if this is if this is your initial condition, this you'll have a behavior like this. But if you change your initial condition to a certain extent, there will be not a slight change in the system. As you can see, the system completely behaves differently, right? So why I'm showing you this is to tell you the importance of the initial concentrations, and also in this case we have not modeled S because S is 0, DSDT is 0, but also the initial concentration of S plays a vital role in determining the behavior of the system. Okay. Now if you take the different null clines, so this is your R null cline here, this is your E null cline. So as I was telling you, like you just put the total or the equilibrium minus E, so something like this, and you have two equations like this. Now these are not like very easy to draw plots, so what you can do, if you want to plot them, you can assume different values of K naught and KR and just check what kind of graph you're getting, right? So for the E and R null cline, if you see, so the blue ones are given by the R null cline and the red one is given by the E null cline. But notice the R null cline depends on S, right? If you see, there is the S factor here. So the R null cline will depend on S, so it will depend on the concentration of S. Just a second. It will depend on the concentration of S. So for E null cline, you only have one null cline, right? This red one, because it does not depend on S. But R will depend on S, and you'll have multiple null clines. So what is the beauty here? So if you notice, for S equal to 10, right, say for the first curve here, for S equal to 10, the R null cline meets the E null cline at one point here, right? Let me take another. Okay. Now if you slightly decrease the value of s from 10 to 6, you see the nullines are nullines are what to say, intersecting each other at 3 different points, here, here, and here. So there are 3 steady states for s equal to 6. There is one steady state for s equal to 10, and if you take s equal to 2, at the end there is only one steady state. So for different values of s, you are switching between the number of steady states. So in this case, s equal to 10 is giving you 1. If you slightly decrease it to s equal to 6, you will get 3 steady states. If you slightly decrease it more, from 6 to 2, you will get only one steady state. Now the thing is, why this is important is basically your stability depends on the concentration of s. Stability and how many steady states you're getting. Excuse me. The stability and how many steady states you're getting depends massively on your concentration of s. Although we are not modeling s because dsdt equal to 0, but still this has a very big role to play when you are trying to check the behavior of the system, right? So s, the concentration of s will determine how many steady states you have in this particular system, right? So yeah, I just wanted to give you this idea. Okay, now you want to check the stability of the steady states, right? So you have 1 or 3 or 2, so this is the 3 steady states wala thing. So for s equal to 6, right, there are 3 steady states, this and this. Okay, so now you want to check the stability of the steady states. So basically ddt and drdt will tell you basically if remember from the phase plan analysis, if you are in a slightly above value or a slightly lower value compared to the steady state, will it pull you towards the steady state, or will you be repulsed away from the steady state? So the drdt and ddt will define the direction of movement in this particular plane, right? On the E nullline, ddt equal to 0, direction of movement is up and down. So basically this is along this nullline, these are your phase fields, basically direction fields, sorry. So at this point it is going down, at this point it is moving up, right? And on the unnullline, the direction of movement is from left to right, so either it goes right or it goes left. And the direction will change. This is a very important thing. The direction of the phase plot or the direction field will change where a nullline is crossed. So basically, if you cross this nullline and move somewhere here, the direction will change, obviously, as we have seen from the stability analysis, because the slopes there will not be the same as the slopes here. So your stability will change definitely, right? Okay. Now, if you draw the whole nullline and all the stability, and basically you have your ddt, drdt and everything. So this is how your steady state looks like. So basically for this particular value of s, you have 3 steady states, 1, 2 and 3. Now try to guess if the steady states are stable or unstable, right? So in this steady state, if you're moving from here, it'll bring you down to here, and if you're moving from here, it'll bring you up to here. So it is this steady state, the first steady state, this part is pulling you in. So basically this part will be a stable steady state. Let me write another. Right? And if you notice at this point as well, this part of the curve as well, this is another steady state. So this is another steady state here. So this is also stable because it is moving down and rightward. So it is bringing you closer to the steady state. Now if you notice this steady state in the middle, it is either pulling you out or pulling you like basically up or down or left or right. It is not converging towards the steady state. So this is basically an unstable steady state. Right. So what have we learned from the whole like model and steady states and everything? So the important things to remember is basically, although your S is not changing, the concentration of S determines how R will behave across the system. If you have also the steady state of R, if you compare it to the if you plot it for the concentration of S, the initial value of R also determines how the system will behave for the steady state of R. So if you have low initial value, this will be a linear plot like this. If you have high initial value, this is a sigmoid. So there is a jump here, there is a sigmoid plot. Right? Next, for different values of S, right? S contributes to the behavior of R. So for different values of S, the behavior of R for degradation and production will change. So I only talked about the production. So production is this one, degradation will be this one. So if you see, it is not getting degraded much and it is producing at a very high level. So what this is telling you is basically the concentration of S determines if you are going to be at a particular steady state for this one, or you are going to be another particular steady state like this one. So the value of S not only determines how R will behave, but it also determines the different steady states, as we saw from here. So if you as we saw from here, so for different values of S, you can have different number of steady states, 1, 3 and 1 again. So in the middle value of S, which is S equal to 6, you will have 3 steady states, which is the maximum, and if you increase it from there, it will go down to 1. If you decrease it from there, it will go down to 1 again. So this is how a mutual repression network works, right? This was a generic example of a mutual repression. Sir has given you many more examples of like positive feedback loops and those stuff. So I thought, okay, I'll tell you a little bit different one. So this is how a mutual repression works. So one such example of a mutual repression is, if I can show you here. Yeah, so this is an example of a this is an example of a bistable system. So why I want to show you this, so this is a MAP kinase pathway, which is a very common pathway that is like studied in basically for your in different types of fields. So in cancer systems also you'll see people studying MAP kinase pathway. In there are different kinds of, say for example this one, oocyte maturation or developmental pathways also, you'll see a lot of MAP kinase being studied there, because it's a very important and a very well studied pathway. So if you see here, so if you see here, so this is a positive feedback loop till here, then it's a negative feedback loop, and then it is basically again doing a positive feedback loop. So if MOS is increasing, it is increasing P42 MAPK, basically MAPK is the protein, and if P42 MAPK is increasing, it is decreasing cyclin and CDC2, which in turn is increasing MOS pathway, MOS protein. So if you can try to think of the analogy from this, so here also you are like producing one thing which in itself you are producing something which in itself is helping you degrade something, right? So this is a mutual repression, and this is also a mutual repression. So you are increasing something which is helping this degrade something else which is doing this. So this is a, sorry, I said this was a repression. So this is a positive feedback loop, if you remember, if you notice. So MOS is getting increased, which in turn is increasing p42 MAPK, which in turn is decreasing CDC2 and cyclin B, which in turn is increasing MOS. So increase of MOS will result in increase of p42 MAPK. MAPK is basically MAP kinase. It's a protein, it's a kinase which helps in phosphorylation of different type of proteins in the signaling cascade. But yeah, this is a positive feedback loop. Now, if you notice, these are the basically the diagrams for describing the system. Sorry. Yeah, this is the diagram describing the system. So if you notice, initially MAPK is low, and then suddenly it jumps up, and you have a steady like what to say, a saturation point. But this is a sigmoidal curve, right? This is a sigmoidal curve. Now if you notice, this part of the region of, sorry, yeah, this part of the region, so if you are moving from this, so this is one curve, this is one curve, this is one curve, and this is the other curve. Like there are 4 curves here. So if you notice this area in this, in the basically the sigmoidal curve, or this area in this sigmoidal curve as well. So these are known as ultra sensitive zones. Why? Because if a slight increase of the concentration of say for MAP kinase, MAP phosphorylation, this compound we are measuring, if a slight increase is done from here, it will directly reach here, which is the saturated state. But if you do a slight decrease from here, it will reach here. So basically these two are stable states, these two are steady states, stable or unstable I don't know, but these two are steady states, and this in the middle is basically the ultra sensitive zone. If you push it up or down, it will basically what to say, it will go and assume that particular steady state. So if it is slightly decreased, it will assume this particular steady state. If you slightly increase this will assume this particular steady states. Okay, so this was basically for forward loop you have these kind of behavior. So this is basically not exactly forward loop, you can call it positive feedback loop. Right, so for a positive feedback loop, this is how your system is going to behave, and you'll going to have certain ultra sensitive zones where if you move slightly from there up or down, you converge into a different steady state than what you were initially trying to do. So if you downgrade it a little bit, it will go in this steady state. If you upregulate it a little bit, it will go to a different steady state. So this is how a positive feedback loop works. So this is an example of the positive feedback loop, where basically they are checking for the MAP kinase cascade for a oocyte maturation, and the behavior of the system is something like this, right? So this is one MAP kinase and like what to say model. Another MAP kinase is basically this one. So this is this paper is this paper, if you go and check out. So here they have described it in great detail, okay, how your different so this is, just let me connect to my ISC email ID, so then I think we can get access to this. Yeah. So if you want the PDF to this, I can send you. If you want to read, where to- yeah. So this is the supplementary. Where do we download the paper from? Here is the PDF. Okay. So Ravi Iyengar is a very famous system biologist. He's one of the earliest ones. So you'll find certain. He's from basically Mount Sinai Hospital. So he has worked a lot on systems biology and related pathways and how you can model them. So what I'll do is I'll download this and send it to you for if you want to read further. This is a very good paper to do that. Oh, this does not go here. Okay, I'll send you the link. See if you can open. If you can't, you tell me your email ID. I'll send it to you there. Okay. Uh, yeah. So here, if you see, just open it from here. So here if you see, they have considered these type of equations, basic chemical reactions, and they have, like, this is a very commonly used Michaelis-Menten model, right? Using these kind of models, what they have done is they have modeled different types of pathways. So this is one EGF pathway, which is basically a growth factor pathway. This is another one gap pathway. This is a CAMK2. This pathway is very prominent in neuronal conduction. Basically, CAMK2 helps you to to basically remember things. The whole concept of memory comes from CAMK2. So these are very interesting pathways if you study them a little bit, and they have modeled these pathways and like shown what kind of activities does this pathway show. And it's a very good read if you want to read that. So NMDAR is another receptor which is associated with basically neuronal conduction and also memory. CAMK2 and NMDAR are very closely related, you see. So I know many of you might not have that level of biology background to understand all the pathways very clearly, but you can see it from a very very subjective way. So this, you consider these as different enzymes or different type of molecules or activators or repressors. Doesn't have to, you don't have to be concerned about what is the function of this particular pathway or what it is doing, but these are very interesting pathways that are present in the that we see in cell signaling pathways. So these are very commonly used pathways in cell signaling and commonly studied as well. So these are very well studied pathways. So if you have a mathematical model like this, and if you can predict something, like say for example for a smaller value of s, as we saw, you have a different steady state. For a higher value of s, you have a different steady state. So these kind of these kind of like inferences that you can make using this model can ultimately help you to design better wet lab experiments. So that is the whole idea for this. So you can get an idea about what is going to happen. And now if you see CAMK2 here, it's here CAMK2. So it's following an oscillatory pathway. So these are very important things. So if you have a feed forward loop, what is your particular type of behavior that you'll notice? If you have a positive feedback loop, what is the kind of behavior you'll notice? If you have a negative feedback loop, what is the kind of behavior you'll notice? If you remember from last class, so just go back to my slides from the last class. Here, if you remember these kind of loops, so these are network motifs, and each of these network motifs has a very distinct behavior of this network motif. So for example, positive feed forward loop will give you this kind of a sigmoidal curve, and there will be an on off. So this is basically a you can consider this as different types of gates of electronics. So basically, if you increase z to a certain extent, after that there is a sudden increase in z. Yeah? So this is basically the double positive feedback loop, where z is increasing x and y, which are like auto-regulating themselves, and x and y are in equilibrium. So it is increasing its x is increasing y is increasing x as well. So this is the concept of memory. So why I'm saying concept of memory and CAMK2 and everything. So if you see this different type of molecules inside the cell, so if you take the case of neuronal conduction in humans, so CAMK2 and what's the other one? I just forgot. Sorry. Yeah, CAMK2 and NMDR, they are very like they have a very well knit system, and they like regulate each other in different forms. So one of the forms is basically this. And why is it important is basically this is what is showing you कि there is a memory of x and y even after z has been reduced. So you increase z to a certain point, and then you reduce it after. So 2 to 4 seconds, so let's assume these are seconds. So 2 to 4 seconds you increase z, but like after 4 seconds you decrease it back to its original form, but that this increase in z is affecting x and y in a form like it keeps the memory of the increase in z. So this is what is called memory in terms of feedback loops. But if you talk in memory in terms of neuronal conduction and memory in terms of brain and everything, this is a very similar kind of a concept is followed in through NMDAR and CAMK2 receptors, where if you increase one of those, the other keeps a memory of that. So that will not get decreased suddenly after this has gotten decreased. So it will keep a memory of that. So what is the memory here? Here it's the memory is a higher basically expression or a lower expression. So similar kind of concepts are used in neuronal conduction as well to basically understand what memory is, because memory is a very it's a very hard thing to comprehend, but this is one of the ways you can comprehend memory. So and this was a positive feedback loop. You have similar kind of so if you have this kind of networks, which basically it's a negative feedback, x is increasing y in turn is decreasing x. So if you have an if you have a network like this, you will find oscillations at different damping levels. So damping and sustainability, I think I'll not cover in this, but if you want to learn about damping and everything, I suggest you go and take a look at simple harmonic motion. So if you simple harmonic motion, it's class 11-12 concept, and damped oscillation and forced vibrations. So let's look at the Wikipedia page only. So here also I'm just moving into physics a little bit, but here it's the equation is given by f equal to minus kx. So f is the force vector, minus k is your parameter, and x is the direction of movement. So this is known as Hooke's law, if you remember. So if you can write f as the rate of change of momentum for a particular element. So this can be dm dt equal to minus kx. So m also you can write. So this is the equation. So f equal to dmv dt. mv is giving you the momentum of the thing. Now this is your equation. So m d2 x by dt2. So you have double differentiated this, and if you solve for x, so m d2x by dt2 equal to minus kx, what you get is basically a oscillatory function. If you remember, cos function is a oscillatory function, so it oscillates from say -1 to +1 in terms of value, and it's a periodic function in terms of like if after every 180 degree it repeats as well. So these kind of functions that that kind of you get as your basically output is actually nothing but a oscillatory function. Now this oscillation in terms of simple harmonic motion, it's there's a uh, there's a spring or something. If you can see, there's a spring or something. This will go on like coming up and down if there is no resistance to the spring, but in real life there is resistance to the spring. So what brings about for the resistance for the string is basically damping. So if your spring is not so like it provides a force opposite to the direction of your oscillation, what it will do is it will create damping. So this is how the damp oscillator will look like. So you'll have another in like f equal to minus kx, and also another term which is telling you about the damping of the simple harmonic motion, which is given by this linear term. So this is how so this is the original oscillation for uh, basically the blue one is the original oscillation, but if you have certain damping, the amplitude will reduce for the oscillation, and ultimately you will be like converging at a particular steady state where basically in terms of the simple harmonic motion of the spring stops bouncing. So a similar concept is basically used here as well. So if this is the system, if you write the equation for the system, it will be a similar system to the simple harmonic motion, but in this case you don't need double differential, you will need only one differential, but the amount of but the basically the behavior, the characteristic behavior of this particular system will be very similar in terms of oscillation. So oscillation, this dotted lines is the original oscillation, and if you take the like solid line, it is showing you the damped oscillation. So after a certain point there will be damping in real life. So this is how a negative feedback loop looks like, and this also we discussed last class. This is an autoregulation and negative regulation. So this is one autoregulation and there is also a negative regulation, and then you can have these in cascades. So for you, what to understand from all this is basically you need to know what kind of characteristic behavior a particular system will exhibit given a particular equation or given a particular motif, like this a negative feedback or a positive feedback or different types of motifs. What kind of behavior does the system exhibit? That is important because like if you don't know that, but basically those kind of questions will come in the exam. Okay, like you are given a particular system, you have to identify if it's a positive loop, it's a negative loop, or it's a feed forward loop, and then you have to identify what kind of behavior will this particular system exhibit, right? So yeah, I think so let me show you another bistable system like this. This is the lac operon in E. coli. So this is the whole network. So it's a little bit messy, but when there is low nutrient level, so E. coli, I hope you know, it's a bacteria. So we grow E. coli in particular in lab using petri dishes. Also E. coli is very readily available all across everywhere there is E. coli is very common. Now E. coli is a bacteria which can use both glucose and galactose as its substrate. So if you think about it, glucose and galactose are quite different kind of compounds, like in terms of their chemical nature of the compound, but it can use both of them as a substrate for its own metabolism because it's an advantage for it. Now this whole mechanism of when it will use glucose as a substrate, when it will be use galactose as a substrate, is given by this complicated network. So when there are low nutrient levels, as in low glucose, this lac I factor, this is the lac I factor here, this lac I factor will repress transcription of the lac A and the lac Y and the lac C genes. So this lac I factor will repress this particular gene. So this particular gene has three elements, LAC-Y, LAC-A and LAC-Z. These help in lactose metabolism, right? This will help in lactose metabolism, shifting away from glucose metabolism. So if there are low nutrient levels, LAC-I will repress the transcription of these three genes. Lactose, allolactose or IPTG, IPTG is this compound, IPTG will bind to LAC-I and relieve the repression. So it's already repressing it. So for example, when you have when you have low glucose, this is getting repressed by LAC-I. Now LAC-I will go and bind to this IPTG molecule, which will relieve the repression of this particular gene, right? And LAC-Y will encode a permease which allows lactose into the cell. Once this repression is taken off and this gene is getting transcribed, what it will do is it will like provide what it codes for is basically an enzyme called permease, and permease will help to integrate lactose inside the system. So if you don't have glucose, you take up lactose. This is the whole process of this. So it's kind of a negative feedback loop here, but this works very well, and this is a very good example of a bistable system. So if you model this lac operon, so this is the whole thing is called the lac operon. Like it is shown in like class 11-12 textbooks as well, if you want to know a little bit more about it. This is a very interesting system. So if you want to model this lac operon, so DLDT, it's basically the lac gene. This is the whole lac gene, which is the DLDT. Change in concentration or change in expression of this whole lac gene is dependent on LAC-Y, because LAC-Y will integrate this permease inside the cell. And also it will depend on L as well. So these are the equations that you can get out from this lac operon, where basically the different expressions are given. L will be the intercellular lactose. LAC-Y is giving the expression of this particular gene, which is integrating the lactose. And these are all constant: beta, gamma, delta, sigma. These are all constant. And EXT is given by the how much external lactose we get. So if you model this, basically another form of the model will be to use d-lac-y dt. So this part as a square of the lactose. So here, if you can see, it is taken as a this is a sigmoidal function, if you notice. So L is taken to the power of 4 because like it helps you like visualize the null lines better, but in general this power is taken as 2. So basically this is how your null line analysis will look like for different LAC-Y and lactose. So for different concentration of LAC-Y, you'll have so for different concentration of L external and L as well, you'll have different type of null line analysis and different types of steady states. So for example, in using this one, you'll have 1, 2, 3 steady states. The other two do not have steady states as well. So these are the type of reactions and everything that how we use this kind of models to understand the behavior of the system. Lac operon is a very good example for that. Okay. Yeah. So let me check one thing. Okay, I think from my side that's all. I hope I have not missed anything. We will discuss certain problems for these in the next live session. When we'll actually focus on certain problems like how you can model this, and like what kind of questions will come in the exam. So I'll discuss these sums as well, like how you can understand and everything. But in terms of the whole syllabus, this is it. On top of this, you have like what to say, you have Sir has given you an idea about how you can use, what to say, uh, uh, databases like Cake. So Cake is one such database. Cake is the Kyoto Encyclopedia of Genes and Genomes. So here you can get information about different pathways. So let's search for the JAK-STAT pathway here. This is also a very well studied pathway. So this is your JAK-STAT pathway. You see here, I am hoping it will open in a bigger window. Yeah, this is the whole JAK-STAT pathway. So it is involved in apoptosis, which is basically programmed cell death, and you can see a lot of lot of different factors that are involved in it. Even the MAF kinase pathway is involved in the JAK-STAT pathway. So for example, you see this these two proteins, cis and SOX. So these go and inhibit JAK and JAK1/3, JAK2, whatever they inhibit. But this again, JAK, in terms of JAK, it will increase the expression of STAT, which will again increase the expression of this particular protein. So here you can see a negative feedback loop. So this is getting negatively regulated, then this is increasing this particular protein again. So this is an example of a negative feedback loop. You can find certain loops like this inside the system. So when you are modeling these are very complex systems. So when you are modeling these kind of systems, you need to identify the small submodules inside the system. A small network, say for example this one is a small network here. How is it behaving? Is it a positive autoregulation? Is it a negative regulation? Is it a feed forward loop, or is it another kind of loop, or whatever? So you can break this whole network into small modules and study these particular elements of this module and get an idea how that particular part of the network will behave, right? So this was just an example, but like what to say, signaling pathways are like this, and these is how it is modeled in systems biology, and we can get different results, right? So yeah, that's all from my side. I'm sorry if I was not up to the mark today, because I'm a little bit ill. So I tried to shift this this session to tomorrow, or like a later part of today, but they did not allow me to do so basically close to the meeting. So I think I've covered everything needed. If there are particular things that you would like to would like me to go over, please have the questions in the for the next live session. So I'll just send you this paper. Yeah, you can check if you have gotten or not. I've sent you the mail. Okay, it is saying that delivery failed. Can you see my screen? No.
No.
Just let me show you once. So it is saying delivery failed because your recipient's mailbox is undergoing maintenance and can't accept messages right now.
Now only I can see your.
But what I tried to do is I tried to send the paper to your mail ID, but it is not going currently for some reason. It's telling ki.
Actually, you are sending through MS Office, I think, right?
Yeah.
So I will share my MS Office.
No. Okay. I have a Gmail as well. I will send it through Gmail.
Also, I have one doubt.
Yeah, please. Yeah, so
Like we are plotting mathematical flow chart, right? Model flow chart.
yeah.
Do you know how to draw that thing, like through any softwares or
Okay,
something?
I once showed you, so I'll show it again. Just a second. There are these plotters which you can use. These are like websites. So if you just want to plot certain functions and stuff, this is the plotter that you can use. You can add multiple equations and everything. You can write it. They'll give
Hello.
give you the
I'm
plot.
not asking about this graphs. I'm asking about mathematical flow charts, like compartment diagrams or something.
Compartment
If we are
diagrams, as in, like, I'm not
we
getting what
are
you're exactly trying to
we
ask.
are plotting this is a model.
Hmm.
We can see some flow charts now. You also included in your
Flow
slides.
charts, in the sense you are telling ki the pathway wala flow charts, how those are depicted.
Your voice is breaking, I think.
Okay, can you just show me what flowchart you are talking about? I'll stop there sharing this screen. Can you just show me once? Then I can understand better.
Wait a minute.
Yeah. Oh, okay. These kind of flowcharts you are talking about. So, uh, primarily,
Yes.
actually, for biological pathways, we have a software called BioRender, but it's a paid software, so you can't use it for free. And the other thing is, these kind of things you can make in PPT as well. So I make it personally in PPT and also in.
Actually, we can make that compartments,
Hmm.
but we have to include these type of equations there, right? I
Hmm.
mean, some these things, beta Si or
Hmm.
something.
Hmm. Hmm.
So it is little bit harder to include.
But the thing is, the softwares that are used to actually, so the kind of these compartments that you see in the papers, those are used primarily, like, there are softwares, paid softwares, which make those diagrams for you when you give certain instructions and everything. So the diagrams that you see there are from paid softwares and everything. So if you want to make it, my suggestion would be use PowerPoint, and like for the particular, say, beta, gamma, and those kind of other numericals that if you require, I think you need to like copy paste from Google or something like that, because PowerPoint won't allow you to just give in the Greek letters. But in general, like, I don't think you need to worry about this, because it's just a representation. You can represent however way you like. But the thing is, the kind of things that you see in papers and everything, those are mostly done by paid softwares. Okay.
Okay, can you suggest the name of that software?
The name of the software is basically BioRender. Right, you can try and look it up, but it's a paid software and it's a little bit expensive as well. So how we use it in our labs is basically, our professor has the software and all of the students use the software for the lab. So it's generally used in that manner. Personal usage will cost you way too much. So yeah.
And also, if we are planning for another session, means try to, if possible, try to make it two sessions.
Two sessions, the issue is, I am only allowed one more session. Like so it's an 8 hour, sorry, a 4 week course. So every week I have to take 2 hours live session, close to 2 hours or whatever it is. So I am allowed total 4 live sessions in this, and an extra live session where I can recap and everything. I'll talk to them if they allow 2 live sessions for this. I'll definitely take them, because it also increases my number of hours, which is a benefit for me as well. So if they allow, I'll definitely take them, but I'm not sure they'll allow, because there's this fixed thing like the number of weeks into 2, that is the total number of live session hours, and extra you can add 2 at the end where you can do a recap of some sort, right?
Also check with the assignments,
Hmm.
because all assignments will be closed on next week.
Okay.
So if
So
possible,
yeah. What I'll do is I'll try to bring up the assignments, and I'll try to discuss those assignments as well. So right now I cannot do that because firstly I do not have access to the assignments. Secondly, since the assignments are still open, I cannot just discuss the answers here. So people will just put the answers there. So that's why I cannot do it right now. I'll do it in that live session. I'll what to say? I'll bring up the assignments and I'll discuss. Don't worry.
okay,
And
that's all.
do you have
Uh,
any preference in the terms of the when I should take the live session? Like as in, like is this time okay, or should I push it a little bit in the evening, or should I do it on Sunday, or like, do you have any preferences like that?
Currently, it's okay for me,
Hmm.
but if it is like 5 to 7, that may be
Okay,
okay, I think.
okay. Then I'll try to put keep the live session then only. Okay. So because I
And
see
before
not many people are
and
allowed.
just before 1 week of exam is will be good, because we also
Yeah,
make
yeah.
some revision.
Yeah. Sir is also of the same opinion. Ki like take the live session just 1 week before. Do not take it like next week or next week. We'll take it 1 month later. So it will be today is 17th. So 1 month later is around like 15th or some time. I'll take the live session. Okay.
Okay, thank you.
Okay. Thank you for joining, and sorry for today. I was a little bit off, but if you have any questions and everything, and like you want me to revise particular topics or anything, just mail it to me. I'll include that in the live session, right?
Uh, okay, I will.
Okay. Okay. Thank you for joining. I'll close it here. Thank you.
Okay, bye. Take care.